Good evening, everyone, and welcome to Kaplan's Inside the Classroom for the MCAT. We are super excited to have each and every one of you joining us here tonight. And I'm really thrilled because you're going to get to really witness some great teaching and get some great study skills and strategies that you can be using right away to start improving your MCAT score. And so we're super excited that you're all here joining us tonight. I encourage you to take lots of notes, to ask lots of questions as we go. That's the point of you being here tonight, is to really kind of get that full immersion into what the classroom experience is like here at Kaplan. And we're going to make this fun and have you learn some stuff along the way as we're going for the next couple of hours. My name is Pedros Manassi. I'm the Senior Director of our pre-health programs here at Kaplan. But more importantly, in my opinion, I am an instructor here for our MCAT programs and actually teach on the MCAT channel with John Linick, who you're going to actually see teaching here tonight. So super thrilled to be working with a great colleague and actually a whole team of colleagues that we have joining us. We actually have a full team of teachers who are going to be answering questions along the way, very similar to how our live online classes operate. So you're going to get a real kind of sense of what it's like to be inside one of our classes. And so that's really kind of part of the experience, is really kind of experience the vast kind of multitude of teachers and expertise that we have here at Kaplan. And so we're excited to be sharing that with you here tonight. Our agenda for the evening is relatively straightforward. We're going to do a little bit of an overview of the MCAT. We're going to talk about kind of how the MCAT fits into the admission space. But then really the bulk of tonight is going to be spent in our metabolism lesson. So this is actually one of multiple lessons that we have as part of the Kaplan MCAT course about metabolism. So we're going to be doing one of those sessions here tonight. So John's going to be teaching that portion. And then we're going to kind of come back and regroup and really kind of go through some different tips for success, things that you should be doing leading up to the MCAT. The MCAT isn't until January, which feels like a long time away, but it's really only 3 months until the next testing date. In fact, registration for the January through June exams opens up in just a couple of days, opens up this coming Thursday. So, really now is the time to be thinking about kind of what it is that you're going to be doing as far as your preparation is concerned. When we think about the MCAT, we think about the importance that the MCAT plays in the admissions process. It's really one of two quantitative factors that go into the admissions process, the other, of course, being your undergraduate GPAs. But we do a survey each year of medical school admissions officers, not advisors, actual officers, deans of admissions, directors of admission, the people making the decision. And we ask them, what's the most important factor in the medical school admissions process? And year after year, the number one response is the MCAT score. In fact, if we were to look at the number 1 and 2 responses, the MCAT score would almost fill up the entirety of the page. Now, it's not to say that the other areas are not important. In fact, medical school admissions is a very holistic process, and that everything is being evaluated about you as an applicant. But the way that you can kind of set yourself apart earlier on in the process is by having a strong MCAT score, because the MCAT really is an exam that not only, it's not just a number, it's really showing your potential for how you'll be performing in the medical school curriculum. And so medical school admissions officers really do kind of take a very strong look at that MCAT score, because it is the one thing in the application that is literally the same for everyone in terms of the measure that you have, or the standard that you've been measured against. That's everybody takes, the MCAT, and we all take different undergraduate courses, we all have different experiences, different personal statements, obviously different life stories. But the MCAT score, that's the one thing that everybody is measured against the same standard. So medical school admissions officers do take it very seriously. Now, the exam, and this is information that's available on our website, it's available on the AMC's website, so I won't bore you with the details, but you can see here kind of what the breakdown of the exam is. But what's important to recognize is that the MCAT, it's a long test. I mean, you're going to be there at the testing location for about 7.5 hours. I mean, there's a lunch break that happens halfway through this exam. So really, when you're thinking about your preparation, it's not just about knowing content, which is a huge part of getting ready for this exam. It's not just about knowing strategies and knowing how to apply the content that you've learned to the types of questions and being able to interpret the questions. There's also an element of building a very strong endurance as you are working and as you're practicing for this exam. So that becomes a large part of your preparation as well. And as you get closer and closer to test day, you'll be taking more and more full-length exams as part of that preparation. But the other kind of fascinating thing about this exam is how interconnected the exam is. I mean, if you look at the names of these different sections, in fact, you look at 3 of the 4 sections, they all have the word biology or some derivative of biology in them because everything is related to the biological sciences. So very, very different from how you have learned some of these subjects as undergrads. You're being tested about physics and chemistry in the context of the biological sciences, right? So it's really an exam that starts to integrate the various sciences. And as you'll see here tonight when we look at metabolism, you'll actually see, and John's going to do a great job kind of pointing these things out to you, seeing how chemistry, which is a subject that you learn in your chemistry classes, really shows up all over the place in metabolism. The same exact principles apply and need to be kind of well understood in order to understand some of those higher level topic areas. So you're going to get a chance to kind of experience that and to see the interconnectedness of all the sciences as they are tested on the MCAT, and kind of the importance of really understanding how all these pieces do fit together. Now, the current MCAT sometimes is referred to as MCAT 2015. And the reason for that is because this version of the test was first offered now almost 4 years ago. The previous version was something like MCAT 1991 because it was offered from 1991 until 2015. So this is, so we call it, you'll hear the term MCAT 2015 float around sometimes, but really at the end of the day, whatever it's called, as far as the scoring is concerned, this is an exam that you are being kind of evaluated against the performance of yourself versus how others have performed. It is a curved exam. I don't know if that's the best way to describe it necessarily, but the idea being you're being evaluated, and your performance is shown how you've performed against all others who have taken the MCAT. Not the others that are taking the MCAT on the day that you're taking the test, but all previous test takers. So a 125 in each of the 4 sections, that is the 50th percentile, meaning that 50% of students performed worse than you, 50% of students performed better than you if you scored a 125. The overall score, the 500, that is the middle score. That is the 50th percentile, meaning if you score a 500, 50% of students have scored better than you. What you have to remember, though, is that for medical school admissions, you're not looking for scores that are in the 50th percentile. In fact, if you look at kind of the average matriculant data that's out there, it's closer to the 85th percentile. Now, that's not to say that you have to be in the 85th percentile to get in. Plenty of students are admitted that are below that number. There are plenty of students who are above that number who are not admitted because, as I showed you earlier, the holistic nature of this exam, and a holistic nature of not just the exam, but of the admissions process is so important to medical schools that there are other factors that will come into play as you move forward. Now, as we look at this exam and we look at the science content that's available on this exam, I alluded to the integrated nature of the test a couple slides ago, but this infographic really kind of shows you how much integration there is. I mean, look at the chemical and physical sciences section, the chem phys section. There is as much biochemistry in that section as there is in the biological and biochemical sciences section. So very, very important as you're preparing for this exam to be really thinking about the integrated nature of all the bits, of all the pieces, and everything be responsible for as you're moving forward as far as your preparation is concerned, and as you're kind of thinking about the sciences and how to use them along the way. Now, the MCAT does have a bunch of skills that it tests you on, and those skills, of course, are content related, but there's also a set of individual skills that you're going to need to incorporate into all of these questions. So the various MCAT skills are things like scientific reasoning and problem solving, experimental data, experimental and research design, data interpretation. Of course, science knowledge comes into play. And in fact, what you'll see, and you'll see this in some of the questions that we do here later tonight, each question is kind of at the intersection of some type of content, as well as looking at this from the perspective of these various skills that you're responsible for on this test. So really something that I want you to kind of keep in mind and think about as we're looking through this and as we're working through the various problems here tonight. So it's going to be quite fun for you to get a chance to see those things and see how all these pieces are really integrated with each other. So with that said, I am going to pass the baton over to my colleague, John Linick, who's going to take us through our metabolism lesson. And so this is kind of the beginnings of metabolism. So we're going to look at glycolysis, which is a very, very important part of biochemistry and a very important part of metabolism. A little bit about John, just before he gets on camera here. John is one of our veteran MCAT instructors. He's, in fact, I think he's been a Kaplan for almost or well over a decade that I know, and is one of our directors of our summer intensive programs, one of our most highest rated instructors that we have, and really somebody that I think you're going to enjoy spending the next hour and a half with or so as he teaches material. Once John is done, we're going to all come back together and we're going to be talking about some various kind of things that you could be doing right from the get-go. But I want you to take lots of notes. I want you to learn a lot of stuff, and I want you to enjoy the next hour and 15 minutes or so of time that you're going to have talking about metabolism. So with that said, John, why don't you come on camera? I'm going to stop sharing my screen so John can start sharing his screen, and we're going to take it from there. I think John's almost with us. There he is. All right, John is here. I'm stopping my screen share, and we should be ready to go.
Awesome. Thank you so much, Petros. As Petros said, we work together quite a bit. We both teach on the MCAT channel. It is a super fun place to teach. And let me start my screen share just to make sure this is all working. This is a slightly different format than we normally use. And so both Petros and I are making sure that we got our tech all lined up. So let me know, teachers, if everything is displaying correctly here. Should be pretty good. But welcome, everybody. So I'm really excited to teach this stuff, as Petra said. There's a lot of things that he said that I'm going to piggyback off of that really connect really well to the MCAT in general. And so one of the big things that he mentioned was how the MCAT doesn't test anything in isolation. And so what we're going to try to do today is not just teach you about metabolism. We're not going to just focus on glycolysis and citric acid cycle and all that kind of stuff. Instead, we're going to connect it to other more complicated, not more complicated, other related processes and concepts such as general chemistry, such as physics, such as, of course, biochemistry in this as well. And so it'll be fun. We'll have a good time doing this. So without further ado, let's get this party started. And also, as you know, we've got a lot of teachers in the room right now. And so I encourage all of you to ask questions, hit us up. I'm going to try to ask you guys questions and throw your answers into the chat that we have there. And we're just going to try to make this as fun, interactive, and engaging as possible, you could say. All right, so let's do this thing. So what we're going to do today is we're going to start off talking about, and actually, let me do one thing real quick on my end just to make sure I have this dialed. Cool, there we go. We're going to be talking about different enzymes that appear in glycolysis. We'll be talking about the regulation of it. One of the big things we'll be talking about is word roots and the most important things that you need to know for the MCAT specifically. And so we're going to go through a lot of things. We're going to go beyond, well, not beyond the scope of the MCAT. We're going to get into the heavier things and we're going to do the lighter things, but we are going to really, really reinforce the essentials, the stuff that you must know no matter what for the MCAT. And that will be a very big part of this. All right, so let's jump in and do it. All right, so here's a little breakdown. As you can see, this is actually something that's missing here. Ooh, this will be fun to fill in. So at the very, oh no, it's not. This is the full thing. We're good. So at the very beginning, we have glucose. And so my first question for you is, when you're going through metabolic processes like this, it's usually easier to focus on the bigger piece things, such as how many carbons do you have? And so how many carbons does glucose have? And I'm actually going to throw a bunch of questions in here. So how many carbons is it? What is do you know the structure of it? Is it D-glucose? Is it L-glucose that matters for us physiologically? And you do need to actually know what the structure of glucose is, that you can draw it out. It is, in fact, D-glucose that we use metabolically. And as folks have said, it is also 6 carbons in its entirety. So it's a 6-carbon structure. And then we're going to break it down into pyruvate, and we'll go further all the way through the Krebs cycle here as well. And so when you look at this, what we really want to think about is in terms of inputs and outputs and the things that matter most. And so what's the whole point of glycolysis? Like, why do we do this? What's the benefit? Like, why would we want to break down sugar. And as you all know, it's because this is how we get our energy. And so we get energy in the form of ATP, as you can see. But notice also that we have to actually invest. That image is a little blurry there, but it says ATP. So basically, we're turning ATP here into ADP. We're going to invest some energy, but in the end, we're going to get even more back out of it. On the actual MCAT channel, we go much further than this and actually get into the electron transport chain, oxidative phosphorylation, as well as fatty acids as well. And so we don't do that tonight. There's not enough time, but we do get into a lot of these related topics. All right, so as you can see, ATP in order to get ATP out as well. All right, so what we're going to do when we go through this is we're going to break it down into the most important steps, the things that are most essential for the MCAT. But before we even do that, I want to challenge you all, because on the MCAT, yes, you need to know the structure of D-glucose. You need to recognize it, whether it's drawn in the linear form or if it's written in its ring form, whether it's drawn as a Hayworth projection or if it's drawn in the actual chair form that we normally see it. That's something that's good to know because you can see it in passages. But there's a lot of other structures here that you don't necessarily need to know the structure itself, but I bet if you know your organic chemistry roots, you can figure it out. So I want to start with one structure that I bet a lot of you know, even though you don't know that you know, and that is this dihydroxyacetone phosphate, DHAP. So it's one of those things that, we see once in a while. You might not think of it as a structure to memorize, but what I want to show you is that it's something you probably already know and already know pretty well. So for example, acetone. Do you know the structure of acetone? Like, could you draw it out if you were given this, if you were asked for it? And this is one of the most common names that you'll see. Things like acetone, acetic acid, formaldehyde. Those are things that you really just need to know the structures of because they use them by name. And so as you know, acetone is otherwise known as propanone. So it's a 3-carbon ketone. And so if you know the structure of acetone, you can figure out the structure of dihydroxyacetone because it's the same thing, just it has 2 hydroxy groups. And so where do those hydroxy groups go? If you were to draw it there, and you can figure it out pretty quickly that, well, there's only 2 places those hydroxy groups can go, and those would have to be on the carbons 1 and carbons 3. And so this must be the structure of dihydroxyacetone. Now going one step further, notice it's DHAP, so there's a phosphate there as well. And so this is an important thing to know about phosphate groups. So one more question. I'll just kind of throw this out there for those of you to think about it before I give the answer. But what kind of functional groups do phosphates tend to bind to? And you'll see this quite a bit in advanced biochem because certain amino acids like serine is a good example, or threonine is another example, have a tendency to bind phosphate groups given their R groups. And so if you know your amino acids, serine, threonine, they have in common that their R groups contain hydroxy groups, which means that it must be at this hydroxy group there that you actually buy the phosphate to make your dihydroxyacetone phosphate. And so what we're doing here isn't saying memorize all of this stuff. What we're saying here is you need to know your fundamentals. You need to know your word roots. And if you know what kind of locations phosphates bind, if you know your amino acids, and if you know your common names like acetone, you can figure these structures out from scratch. And so that is, in fact, the structure of DHAP, dihydroxyacetone phosphate. So kind of big on the top of the page there. I want to go one step further. We're not going to go through a bunch of different structures today. That's not the goal. But I want to start off by doing this because people would often think of this as the most advanced version of knowledge of these things. But what I'll show you is that it's not that it's stuff to memorize. It's not advanced knowledge. If you know your fundamentals of organic chemistry, which you're usually required to take before you even take biochem in the first place, you can figure all this stuff out as you go along. And so I'm going to go through the next structure here. So we talked about DHAP. Let's talk about G3P as well. These are ones that you can infer what the structure is given fundamentals. Now, things like glucose, glucose-6-phosphate, fructose, glucose and fructose are the 2 big structures there. You got to just kind of memorize. The name doesn't tell you what it is. It's a named molecule. But these you can infer if you know some root structures that relate. And so if you look at glyceraldehyde, you'll notice that it has this glycer root. And so is there any other structures that have that glycer root that we know, where you see glycer something, especially when it comes to metabolism? And we do see a lot of glycerol. Glycerol we often see in the context of fatty acids, so triglycerides. the backbone that builds our stored form of fat, those TAGs, triacylglycerides or triglycerides, it means the same thing, is 3 alcohols bound together in a 3-carbon chain. That's a structure that you definitely need to know. Just like acetone is a common one, glycerol is another common one. So 3 carbons, as you can see, 3 hydroxy groups as well. And so glycerol is this structure, thus glyceraldehyde. As long as we know our fundamentals of orgo, we can infer this structure here. So then instead of having an alcohol on one side, you have to just have an aldehyde instead. So you just form a double bond to oxygen, and now it is an aldehyde. And then there you go, glyceraldehyde. And same as before, there's a 3-phosphate. So just as last time, we do need to know that phosphate groups have a tendency to bind to hydroxy groups, especially in things like amino acid chemistry. And so you just throw your phosphate group on here as well. All right, so now please do not write these down as structures to memorize. That is not the point of what we're doing here. What we're trying to do here is that your goal on the MCAT, because you are definitely going to see things that you have not seen before. Every single person who takes the MCAT sees some info that's brand new. And what your job is on this exam is to synthesize, to apply it, to learn about it, and to figure out what it's talking about. And if you know your word roots, you can figure out a whole lot of stuff. That's kind of what we're going for here. All right, so we're done with structures for now. We'll do a little bit maybe later on when we get into the TCA and talk about those structures. But for now, let's get some more focus on things like enzymes and word roots and see what that looks like. All right, so we're going to start at the very beginning. And also one thing as we get into this that I do want to just point out, when it comes to glycolysis, you do not need to memorize the entire glycolytic pathway for the MCAT. We want to focus on things like word roots, on things like enzyme categories, rate limiting steps, and irreversible steps in that order. And so there's one more thing that we should say about word roots before we get too deep into this. And that is just the name itself. It's called glycolysis. And so the name itself tells you what we're going to be doing. And so what does that name mean? If you just read glycolysis, right? Glycolysis. Like if you were to define that, given just like the name there, as you know, glycosylating something. Glucose is a good example. It means sugar. So glyco refers to sugar, and lysis, as you know, to lyse something is to break it or to chop it or to cleave it or whatever you want to call that. And it's exactly so. Glycolysis basically means breaking up sugar or breaking down sugar. It all means the same idea. So that is your glycolysis. All right, so that's a word root there. We're going to do the same thing for a bunch of other stuff that we run through. We're also going to focus on enzyme categories. In fact, of everything that we talked about in metabolism is really going to be enzyme categories. That'll be a very important part of this. All right, so you got to know those, and we'll talk about them here. All right, so step number 1. So if we know this, awesome, but what is the first step? What is the first enzyme in glycolysis? The very first step, and there's actually a couple different reasons that we do this. So you've got your glucose, we turn it into glucose 6-phosphate, and the enzyme that does that is hexokinase, or you can call it glucokinase in the context of the liver. We get more into that in other like channel classes that we do, where we talk about gluconeogenesis and the organs that can do that. But hexokinase is a classic example. So once again, word roots, right? Hexo, as we know, means 6. Notice it's a 6-carbon sugar, as you said. Glucose is 6 carbons, fructose is 6 carbons. And then kinase, very important enzyme category. So what do kinases do? Like, what is the purpose of a kinase? This is super important. You see a lot of kinases in the context of metabolism and just in physiology in general. But they are a type of transferase. So if you want to get broad category, there are 6 main categories of enzymes. One of them is transferases, and a kinase is a type of transferase. Specifically, as we've already talked about a whole bunch, it transfers phosphate groups. And so that's what hexokinase does, is that it transfers a phosphate group to glucose. Notice specifically, it's taking it from an ATP, that's adenosine triphosphate, and then adding that one phosphate, thus turning it into ADP, and adding that phosphate directly to your glucose here. So that's the whole idea with hexokinase. Now, additionally, there's a couple other things here, not just memorizing what this thing is, but figuring out why it has certain things here, like this magnesium. So magnesium is bound to an enzyme. And another good question, what do you call it when you have some kind of inorganic helper molecule like this that's bound to an enzyme, and it helps that enzyme function more efficiently? And so there's 2 main classes that we'll see for these. There is coenzymes if they're organic, and cofactors like this magnesium here if it's inorganic. So remember, inorganic means it's non-carbon based. Organic means it's carbon-based. And so things like magnesium and other metal ions, those would be cofactors. And so one more kind of connecting the dots together here, notice that it's really important for kinases to have divalent cations, right, positively charged like magnesiums, because if you were to draw out a picture of an inorganic phosphate, well, I guess we draw the phosphate here at the end of ATP. We'll draw like the full chain. So imagine that this is kind of going through the rest of ATP. But if you draw your actual phosphate, you'll notice that the end of your phosphate chain actually has 2 negative charges on it. Both of these oxygens would have negative charges. Thus, if you want to transfer that phosphate group, it's a whole lot easier if you've got something with a bunch of positive charge on it to actually transfer the phosphate groups. So that's the whole idea with your divalent cations. You're going to see a ton of magnesium in all the examples that we do today. So it's just one example here with hexokinase. All right, so as you can see then, if you're going to transfer a phosphate group to glucose, that means glucose now has a charge on it. So why is it also beneficial to add a phosphate group, putting that charge in it, other than to start the whole oxidative processes? Because, part of it is, yes, you want to oxidize it. Adding phosphate groups is a way to do that. We're going to do this a whole bunch as we go through the whole process. But there's another benefit. And the other benefit is that it traps glucose inside the cell. Remember, when things have strong positive charges like that, like we have for glucose here or for phosphate groups, that means that it's no longer permeable to cellular membranes. And so once you phosphorylate it, glucose is effectively stuck. It's not going anywhere. It is where it is. It's inside the cell. So that's helpful, as well as, of course, starting the oxidative process. All right, so now this is, as we said, it's a very important enzyme. It is one of the irreversible enzymes, and you want to know that for sure. Those are the steps that tend to be most tested. So we said we hydrolyze ATP, right? Add one of the phosphate groups to glucose. It's an irreversible step. And as we said before, that traps the glucose inside the cell. All right, so real important parts there to definitely know about what this first step is. All right, so remember, first step of metabolism, it's irreversible. And I do challenge you all to remember which steps actually are irreversible in metabolism, in glycolysis specifically, because we have another process that's similar but backwards that we call gluconeogenesis. And so the thing is, if you've got an irreversible step here, then you cannot use hexokinase for gluconeogenesis. You cannot reverse this. You actually need a different enzyme for gluconeogenesis. But all the steps that are reversible, gluconeogenesis uses the exact same enzymes because they're reversible. So basically just redos those steps, but in backwards. And remember, I mentioned this before, but gluconeogenesis happens primarily in the liver. It actually happens in the kidneys too, but most important for the MCAT, it happens in the liver. It's your main metabolic organ. All right, now here's another image. And so what does this mean? When you see that little negative sign kind of going back, this is a very, very, very common step in most of metabolism. Well, not just metabolism, physiology in general, where you'll see some kind of a product, right? You could say this is the reactant, glucose, that's your product. And whenever the product slows down a reaction, as you can see here, that is known as a negative feedback. Nice, yeah, so it's negative feedback. And so that's just an example here. We're going to see a bunch of different arrows here that show feedback. I'm going to go through them fairly quickly, but just be aware throughout all of these processes, you're going to see these kind of annotations over and over again that are just showing the products of a process often inhibit the reaction itself. I'm going to slow it down. That's called negative feedback. You'll see tons of examples. All right, so that's the first part right there. Glucose getting turned into glucose 6-phosphate. We are actually in these slides going to skip the next step here. But I want to mention it briefly because it's another class of enzymes. And we're going to really try to focus on these enzyme classes today. So far, we've only introduced a kinase, and it's a transferase that transfers a phosphate group. This is turning a glucose 6-phosphate into a fructose 6-phosphate. Thus, you're turning one isomer into another isomer. Those are known as isomerases. Specifically, these are structural isomers. They've got different functional groups. And actually, do you guys know what is the main functional group or the highest priority functional group on fructose? I can actually go draw glucose real quick. So glucose has an aldehyde on one end, and then it has a bunch of OH groups down the side. Actually, I won't draw them all, but basically there's a bunch of OH groups all going down your glucose. This is actually only 5 carbons. You'd add another one. I ran a room. What I care about here, though, is that you have an aldehyde as your highest priority group in glucose. When you isomerize this to fructose, it actually turns your aldehyde, nice, not from an It's no longer an aldo sugar. You call it a keto sugar. So what it's doing is it's moving that carbonyl group there from carbon number 1 to carbon number 2. And once again, I'm not drawing out the full structures there. It's kind of a big structure, but that's the idea. So moving your aldehyde into a ketone, that is the big difference. All right, so that's an isomerase. You'll often see isomerases written as mutases as well. So either mutase or isomerase, that's the same idea. Just so we said, kinase is a type of transferase for the first one. So what we're going to do next is we're going to focus on this third step, turning fructose 6-phosphate into fructose 1,6-bisphosphate. And you can already infer what kind of enzyme this is. Because notice, if you're going to be adding an ATP once again, and you're starting off with fructose 6-phosphate, and now you've got 1,6-bisphosphate, so it's got 2 phosphates on it. That means you're transferring another phosphate group. And if you're transferring another phosphate group, that means it's another example of a kinase. So this is, in fact, one more kinase. Now, this is a very important kinase, though. In fact, I would say of everything, this is the most important kinase. Any guesses why? Of all steps in metabolism, or at least glycolysis, this is the most important one to memorize. It's going to be a common theme. First, as we said, you got to know your word roots, your name roots, your enzyme categories. You got to keep track of your carbons, and you need to know the rate limiting steps. This is the rate limiting step of glycolysis. And so it's turning fructose 6-phosphate into fructose 1,6-bisphosphate via phosphofructokinase 1. Yeah, so PFK1, that is the enzyme. Definitely memorize that one. Super important. You got to know it. Gotta know it. All right. All right, so let's talk about a couple of things. So once again, ATP is hydrolyzed. So we're going to phosphorylate your fructose 6-phosphate. It is also irreversible, as your rate limiting steps will tend to be. And that's, as we said, it's the rate limiting enzyme. So super huge. So since this is the rate limiting enzyme, you want to think about what kinds of molecules would speed up the rate of this. And so the classic thing is if you're talking about glycolysis, you're talking about making energy. You're talking about your amount of ATP. So if you are really short on ATP, that means you have a lot of AMP, that's adenosine monophosphate, or ADP, adenosine diphosphate. And so that's the idea. If you have a lot of ATP, then it's going to slow down. If you have a lot of ADP and AMP, right, adenosine mono, 1 phosphate, ADP, 2 phosphates, that's actually going to speed this thing up. All right, so that's the idea. And that's just kind of, I would say, classic feedback, though, right? If the whole process of glycolysis is to make ATP, then it would make sense that ATP would inhibit the rate-limiting enzyme. So kind of straightforward stuff there. Now, where things get a little bit more complicated, we get more into this into the other channel classes where we talk about, it's like we have more metabolism that focuses on hormones, like insulin, glucagon, fatty acid stuff as well. But in the context of the liver, we actually do have an enzyme called PFK2. That's phosphofructokinase 2 instead of PFK1. That makes, as you can see, fructose 2,6-bis. Now, this might seem like, oh, you're basically doing the same thing, but fructose 2,6-bis is not part of the glycolytic pathway. It has another function. It actually serves to upregulate phosphofructokinase 1, PFK1. So this is where insulin comes in. So the whole idea of insulin is if you have a bunch of ATP, but you ate a bunch of food and insulin's been released, you don't want to slow down metabolism. You just ate a bunch of food, you got to use it. And so in the context of insulin being around, even if ATP is high, doesn't matter. You're still going to crank up your metabolism because sugar is available. And so you can make it then to store it instead of just to use it right then. Not necessarily to make ATP at the moment, but to store for later, which you still would do this step. So that's the idea. That's where insulin comes in. As I said, we get way more in depth with that in other channel classes, but this is just your brief little introduction of how insulin fits into glycolysis. All right. So once again, remember, this is the rate limiting step, super high yield. You definitely need to memorize that. Also, big takeaways that if you ever see kinase, remember kinase is a type of transferase. It transfers a phosphate group specifically. All right, and so once we make our fructose 1,6-bis, we then effectively chop it in half. And these were the two enzymes that we started with, glyceraldehyde 3-phosphate, as well as DHAP, dihydroxyacetone phosphate. So now for the purpose of this class, we're just going to say, and this is what most commonly happens anyways, that all of this dihydroxyacetone phosphate is going to isomerize into glyceraldehyde 3-phosphate. And so you can kind of say you get 2 equivalents of G3P. And you actually do often see this written as just G3P. That's glyceraldehyde 3-phosphate. Cool. All right. And as I said, we don't do alternative pathways in this class, but we do in other classes. All right. Let's talk about going on. So now what we're going to do here is we're just going to pick up effectively from your glyceraldehyde 3-phosphate and keep going on from there. All right, let's do it. All right, so yeah, let's do the next step. So this is going to introduce our next main class of enzymes as we go through. But we're going to turn glyceraldehyde into 1,3-bisphosphoglycerate. This is actually one more thing to point out too as we go through this. Another word roots, and I'm really going to try to focus on word roots as much as possible as we go through all of these processes. But your glyceraldehyde 3-phosphate is turned into 1,3-bisphosphoglycerate. You'll often see that difference in dye. Like, for example, if you do ADP, I'll draw a real quick version, a very rough version of ADP, but this would be your base up here. That's like your aromatic ring. So you have your phosphate groups actually all stacked in a row. So whenever you see them all stacked in a row, like ADP, then you call it, notice, diphosphate. The difference is if you're going to talk about bisphosphate, this is where the different word root comes in. Instead of di, they call it biz. Biz refers to phosphate groups that are not linked together like it was with ADP, that they're on different sides of the molecules. And so you actually have your, I'll just put P for phosphate. You guys know what I'm talking about. Your phosphate groups on other sides of it. It was the same thing when you saw your fructose 1,6 bisphosphate. Same thing. Bis means it's on opposite sides. They're not actually stuck together. So that's the difference. All right, so cool. All right, so we're going to keep doing that, kind of going through everything, focusing on word roots, big picture stuff, and main takeaways. And this next step is also super important because it introduces our next class of enzymes. So what do you call this class of enzyme? Notice that you're going to turn your NAD plus into NADH. This NADH is known as a high energy electron carrier. Very, very important for a certain process. Do you guys know what process actually utilizes NADH? We don't get really into it today, but once again, it's on other channel classes. There's a lot to talk about in metabolism. It's a whole semester, as you know. So we're just doing a small chunk today. Excuse me. Yeah, so this is known as a dehydrogenase. So this is glyceraldehyde 3-phosphate dehydrogenase. So to get more specific, a dehydrogenase is a type of oxioreductase. I'm kind of giving you guys the main categories of enzymes. We've already covered 3 of them. So first was a transferase. Our example was the kinases. We've seen a lot. The other example was an isomerase. The one we saw was mutase. Now we've got our third. It's an oxioreductase, and this is a dehydrogenase. It's a type of oxioreductase. So your shortcut for metabolism is anytime you see any enzyme reducing, remember this would be the oxidized version, this would be the reduced version. If you ever see an enzyme reducing one of these electron carriers from NAD plus to NADH or from FAD to FADH2, that means it is a dehydrogenase. That's the kind of stuff to focus in on first when you're learning these things, because if you know your classes, the MCAT's a multiple choice. You can often eliminate most of the answer choices if you just know your enzyme categories and use some critical thinking skills. So that's going to be our real main focus through most of this stuff. Same thing here. So it is a dehydrogenase, right? That's a type of oxioreductase, as we said. And then what else would be good to mention here? Oh, actually, so one more thing too. And this is a very, very, very high yield topic. So as we said, if you're going to be reducing the electron carrier, then by definition, you must be oxidizing the substrate. Super high yield idea, especially metabolism. You never, ever reduce anything without oxidizing something else. Like oxidation and reduction go hand in hand. You cannot have one without the other. And so one of the easy ways to solve questions actually on the MCAT is they might ask you what happens to some substrate. And you're like, gosh, I don't know. That's a really complicated structure. I have no idea if it was oxidized or reduced. You can look at the other species, and you'll often see other species. Like a good example is you'll see a reaction where you see like I2, and then the product side, it's HI. So from this, let me know, what happened to iodine? Was iodine oxidized or reduced in this process? And so this isn't as bad as looking at macromolecule, because if you know your oxidation states, you know that, well, iodine all by itself, that means that it must have had an oxidation state of 0. Sorry, it looks like a 6, that's a 0. And when it binds to hydrogen, well, hydrogen is +1, unless it's a hydride, that's unique though. So then iodine must be -1. Thus, you can say iodine was reduced. So this would be an example of a reduction. So then what other species you had in this reaction must have been oxidized. And I know this is an unbalanced reaction. I'm just showing you a little chunk of a reaction. This is definitely not balanced. You need other species as well. But it's the idea that you can find easy chunks of reactions, like looking at just the iodine. Once again, that's of course not a balanced reaction. Just trying to give an example here. All right, so we'll erase that. All right, so there's a couple other things. So as we said, NADH is generated. And so, let's get advanced. Question for you all. Do you remember which complex NADH goes to? We're going to get that specific. And this is stuff that you want to know for the MCAT. AMC definitely asks questions about individual complexes of the electron transport chain. And so NADH is one of the principal electron carriers that you'll see. It's made both in glycolysis. We make tons of it in the TCA cycle as well. Gotcha. And so it is, it goes to complex 1. Yeah, so this goes to complex 1 of the electron transport chain. Cool. Cool. Awesome. Sounds good. Great. All right. And so as we said, that's a high energy intermediate, and it is in fact reversible. So this is not one of the steps that is irreversible, which means that we don't need to memorize it. The main thing that you need to know is that it is a dehydrogenase. That means it's an oxioreductase. That's mostly it. All right. Cool. Let's do the next one. All right. So then if the next step here, notice once again, and we're going to kind of go through these more quickly. These are actually the reversible steps. So we will go through it a little bit faster. But so this is 1,3-bis-phosphoglycerate to 3-phosphoglycerate. So notice what we did here. We moved a phosphate group. And what do you call the enzymes that move phosphate groups? This is just another example of an enzyme that's moving a phosphate group. The cool thing with this one though is that you're adding to the still a type of kinase. This is phosphoglycerate kinase. Right. All right, so ATP is generated, as we said, and we're generating ATP because you're moving the phosphate group to the, basically from the molecule. Nice work. And so, basically we're going to generate ATP. In so doing, we are going to, do an example of, that's the idea, is that you're going to see a couple different types of phosphorylation that occurs on the MCAT. There is oxidative phosphorylation and there is substrate level phosphorylation. So this is a good example of substrate level. Do you guys know where we also see substrate level phosphorylation in metabolism? There's another part too. Gotcha. Yeah, so it's also in the Krebs cycle in the TCA. So yeah, we see substrate level phosphorylation in glycolysis. We also see it in the TCA. And also, I'm going to keep using words like that. So there's the TCA that stands for the tricarboxylic acid cycle. You can also call it the Krebs cycle. You can also call it the citric acid cycle. It's all the same thing. All right. So this is reversible, as we said before, so we're not going to stress too much about it. The main thing we want you to know here is that it's a type of it's another kinase. So if you see kinase, think transferring phosphate groups. That's what matters most. All right. All right, so notice that we're going to skip all of this stuff here because these are all examples of reversible steps, and we're focusing on the irreversible steps. We're focusing on the ones that have main enzyme categories. So those are just more examples of isomerases where you're moving around phosphate groups but not adding anything net new or taking anything away. That's what matters most. All right. So let's do the last step. So last step's also important because, well, first of all, what kind of enzyme do you think this is, given what we're doing here? Since we're removing a phospho group from a molecule, we are adding it to ADP, thus making ATP. It must be another example of a kinase. Yeah, so kinases are effectively, once again, there are types of transferases that transfer phosphate groups. I know I've said that 1,000 times, but that's what matters most. That's the highest yield thing, and that's what you really need to be comfortable with. So yeah, kinase, you got it. So another thing that's important about this one is that we generate ATP. As we said, it's still substrate level phosphorylation, right? We see substrate level phosphorylation in glycolysis and the Krebs cycle. Later on, after the electron transport chain, that's when we get our oxidative phosphorylation that we'll talk about later on. Not today, but in other classes. All right, and once again, this is irreversible. That means that you've got to know the steps. So pyruvate kinase, that is an irreversible enzyme, which is going to really make a difference once you dig into, going to make it into gluconeogenesis. Got it. All right, so that's the big picture. That's all of it. And so this is actually a neat little thing. I like to think of this. So this is called feed-forward activation. The example of this would be to think of the I Love Lucy episode. If you guys remember that, that might be a little too old at this point, where they work at Lucy and her friend, whatever friend's name was, work at the candy factory where they're basically working too slowly. So all this candy's coming down the belt before they have time to wrap it. So they just find themselves, oh, it's Ethel, thank you. Yeah, so Lucy and Ethel are working at the candy shop. And so they find themselves just trying to eat all the candy because they can't keep up with it. Since we're doing glycolysis, right, sugar breakdown, it's a very good example. It's the same thing. And so what happens is if you have a bunch of fructose 1,6 bisphosphate in your cell, that means that you're going through a bunch of sugar. You are metabolizing like crazy. And so you need to make sure that if fructose 1,6 bis is in process, you want to speed up the very last step of metabolism. That's basically Lucy and Ethel wrapping up the candy to make sure that you don't have a bunch of backlog. And so by speeding this up, remember, this is the irreversible step, you can clear out the conveyor belt, so to speak, so that way you have a bunch of room for more candy coming down the line, which is what's implied if you've got fructose 1,6 bisphosphate. So that's kind of the idea. All right, so yeah, random I Love Lucy reference. All right, and yeah, thanks for knowing Ethel too. I appreciate it. I work with great teachers. They know things like this. All right, so that's a big takeaway. Once again, I want to review the most important steps. Remember, kinases add phosphate groups. Dehydrogenases are oxioreductases. They move around, in this case, a hydride to form a reducing agent. And also we had our isomerases there as well. So we've got kinases, we've got isomerases, we've got dehydrogenases, and the most important steps to memorize are, of course, the rate limiting step, as you see here with a star, as well as the irreversible steps that we already covered. These guys right here. All right, so let's talk next. Let's talk big picture. Let's talk about what we're getting here. So we said that we have two equivalents, right? Remember, you're getting 2 times the equivalence of G3P, which means you net get actually 2 ATP, right? You ended up using 2, you got 4, so you net gain 2. You also get 2 NADH as well as 2 pyruvates. All right, so now before we move on, we're going to cover a bunch more here. We want to give you guys 2 different questions to work through, and then we'll jump into the TCA, right? The citric acid cycle, the Krebs cycle, the citrate cycle. It's got lots of names. We'll talk about all of them, but we'll have you guys work through these questions first. So Chris, if you got that poll already, let's give everyone a poll so they can work through this question. We'll give you like a little less than a minute or so. We'll see how folks are doing, and then we will discuss. Awesome. Let me know the results when they're coming in. Awesome, thanks. All right, so we got results in. So it looks like we've got most of us going for the right answers. Really nice work. And so we talked about this before. Actually, there's one thing we didn't mention before. We definitely know that ATP will slow phosphofructokinase. We mentioned this before kind of briefly, but that if you have either AMP, adenosine monophosphate, or ADP, adenosine diphosphate, that's actually going to speed up this reaction because it means we don't have enough energy. The thing that does not matter is inorganic phosphate. And so that's the thing. The ADP-ATP ratio definitely affects it. Inorganic phosphate does not. Inorganic phosphate's actually under a completely different control system. That's in the context of things like your calcitonin, parathyroid hormone. Calcium balance is actually directly related to your phosphate balance because the way that we store calcium in our bones is as calcium phosphate crystals. With, it's a hydroxyapatite. There's some hydroxy in there too, but mostly it's calcium and phosphate. And so those two tend to get lumped together. And so yeah, that's a different control system, but ATP, ADP, and AMP are all going to affect the rate of your metabolism. All right, let's do poll number 2. So we'll clear out the polls there. Let's do a second one. Once again, we'll give you about a minute or so to solve this. Once we have a bunch of answers in, then we will discuss. Awesome. Thanks, team. All right. So this is one of those moments that it's just straight memorization, and we don't love this kind of stuff, but it does show up in the MCAT once in a while as well. Well, quite a bit. But, a lot of what they're trying to do on the test is exactly as Petros was saying. It's not just memorize things; it's apply it. It's critical thinking. It's analysis is more important. But yes, you do just need to memorize stuff as well, as you can see. So this is a good example, and this is stuff that you want to know. And we are actually going 80% correct. Nice work. Phosphoglycerate kinase is reversible. So hexokinase was step 1, phosphofructokinase was step 3, pyruvate kinase was step 10, the final step. So notice that this actually works out pretty well because the irreversible steps, it's the first and the last, as well as the rate limiting. Like that's not that much to memorize for glycolysis. The first step's irreversible, the last step's irreversible, and the rate limiting step is irreversible. The rest of them are reversible, and phosphoglycerate kinase is one of those examples that is reversible. Cool. All right. So Let's move on to part 2. So part 2 of this class is going to be all on the citric acid cycle, otherwise known as the Krebs cycle, after the scientist who first kind of completed the annotation to figure out how it worked. It's also known as the tricarboxylic acid cycle, or the TCA cycle. And so one other thing that I want to point out here, before we get too deep into the, the structures and the reactions here, is that this first step here is actually not part of the TCA. This is actually its own step. And so what is this called? Do you actually know when you remove a carbon dioxide from pyruvate? It's the name of this whole process. This is a common name that you'll see a lot in orgo, but usually if you add a carbon via carbon dioxide, we call it carboxylation. If you remove a carbon via carbon dioxide, we call it decarboxylation. And so this would just be pyruvate decarboxylation. This is actually separate than the TCA. So when you think of the Krebs cycle, the TCA cycle, that's the bottom circle that we have here. The top is actually separate, yet they both happen in the same place. So where is it that all of this stuff is actually happening? Like in what part of the cell, you could say, is all of this stuff happening? To get more specific, in what part of the mitochondria is this happening? And so this all occurs in the mitochondrial matrix. So mitochondrial matrix is where you see pyruvate decarboxylation, as well as the citric acid cycle, as well as beta oxidation as well when you talk about fatty acids. We won't get into that today, but that's another process that happens here. Cool. All right, so one more thing I want to mention before we get too deep into the specific steps here in terms of just big picture concepts, as we've been trying to say in trends, is notice that a lot of these, actually, let's just do citrate as a good example. So remember, people call it the citric acid cycle, yet here it's shown as citrate. This is a common system of nomenclature. Whenever you have a carboxylic acid, it ends in ic acid, right? Oic acid. Yet whenever you have its conjugate base, which is what we have here, it's not ic acid, right? Once you lose that proton, now it's the conjugate base. Then it's known as the ate conjugate base. So citric acid has the citrate conjugate base. Acetic acid has the acetate conjugate base. Pyruvic acid has the pyruvate conjugate base. It's all the same idea. And so in physiological conditions of your matrix, all of this stuff is going to be deprotonated. Like you never actually see citric acid in the citric acid cycle. Like it doesn't really make sense that we call it the citric acid cycle. It's always in the form of citrate, the conjugate base. So it actually make a bit more sense if we called it the citrate cycle. Yet once things are named, they kind of stick around, as I'm sure you know. All right, so that's a couple background things. Let's keep this party rolling for now. So as we said, this first part is called pyruvate decarboxylation. The first step, of course, is the formation. Well, it's not the first step of the Krebs cycle. It's the step that leads up to the Krebs cycle. So what happens before the Krebs cycle, you could say. All right, so once again, though, this happens in the matrix, very important to know. And so this is also not just an enzyme that does it. It's an entire complex of enzymes. There's a bunch of enzymes that function in this. It's known as the pyruvate dehydrogenase complex. You'll often see it written as PDC or sometimes as PDH. So yeah, PDH or PDC, that is the same thing. PDH, PDC, same, no biggie, no difference. Also notice that there are a whole bunch of enzymes here, as well as a whole bunch of cofactors and coenzymes as well. So you've got a bunch of different vitamins and other coenzymes and things like that, and magnesium. Don't stress about those. Those are not super high yield. You don't need to worry about it. But do be aware it's a big complex. Thus, there's a bunch of different cofactors and coenzymes. So now notice that's because several reactions are happening. You're going to be transferring a CoA group. You're going to be reducing an NAD plus into NADH. And you're going to be removing a carbon dioxide. And so there's a lot of different steps that happen in this enzyme complex. All right, so as we said, this is happening in the mitochondrial matrix. It does, the reason it's named dehydrogenase is because we make NADH. And remember, NADH, that's the first ingredient of the electron transport chain. So that's going to jump into the ETC. It also is basically making acetyl-CoA. And acetyl-CoA is very important because it's the principal substrate of the citric acid cycle. All right, so now when we draw acetyl-CoA, I want to get a little bit more into some orgo here. As I said, we want to connect the dots together in a bunch of ways. So acetyl refers to a 2-carbon carbonyl. So that's an acetyl group right there. You might even remember back to your orgo, in college days, like Friedel-Crafts acylation, right? Where you're actually adding one of these groups here. It's similar root there. This is acetyl, so it's a 2-carbon acyl group, but same idea. But acetyl-CoA is actually bound to a sulfur, or the acetyl group is, with a CoA group. So what do you call these functional groups when you've got a carbonyl carbon that is directly next to a sulfur? So it's really similar to this group, where if you had an oxygen bound to it. And so if it was an oxygen, we call it an ester. If it's bound to a sulfur instead, it's the same thing, but you just call it a thioester. You'll actually see that quite a bit where you see that thio root. Thio always refers to a sulfurous compound. So you can see a thiol in the context of a sulfur with the hydrogen on it, like an alcohol, but with sulfur, or a thioester here as well. So same idea. So do be aware of those thio groups. You'll see lots of sulfur. Such as, super common one of the very important amino acids you need to memorize is cysteine, because it has a thiol. It forms your disulfide bridges. And so you see a lot of sulfur being important. Remember, it's very similar to oxygen because they're in the same group. That means the same column on the periodic table. All right. So as we said before, remember we start off with glucose that was 6 carbons. We chopped it in half. Thus, we got 2 pyruvates that were 3 carbons. We just lost a CO2. Thus, your acetyl-CoA is now down to 2 carbons. All right. So once again, we've got positive and negative inhibition, or feedback, you could say, where you're going to speed this stuff up if you have a lot of, if your cell is short on energy. So if you have a lot of ADP or NAD+, that's basically saying you have a lot of reactants, and adding reactants speeds up a reaction. Same thing. It's kind of like Le Chatelier's principle. What's different here is that we didn't talk about calcium yet. So can you think of any processes that are very energy demanding that use calcium? Because this is a good reason why, if you have a whole bunch of calcium around, it is going to speed up not only your pyruvate dehydrogenase complex, but actually a whole bunch of other ones in the Krebs cycle as well. And that's in the context of muscle contraction. So muscle contraction, right? Your sarcoplasmic reticulum stores a whole bunch of calcium. And once you actually have a contraction, it's due to the release of calcium that binds to troponin, moves tropomyosin out of the way. Then you get your actin-myosin cross bridge, right? We just said a lot of stuff there. We're not going to cover all the bio of it, but that's the idea. Calcium is necessary not only for muscles, but also for things like neurotransmission. You need calcium for your vesicles to fuse with the presynaptic membrane, which is a very high energy process as well, right? Thinking's hard. And so lots of processes that require a lot of effort take calcium. So calcium tends to speed this stuff up as well, because it means you need energy. Whereas products, as we said before, NADH or ATP will do the opposite. They'll actually slow this thing down. If you got a lot of products, processes slow. All right. So we don't cover this in today's class, but in the context of our other metabolism classes, we get more in depth, and we talk about fatty acids, beta oxidation, ketone bodies, ethanol, stuff like that. But the idea is, and this is a real important idea, is that pretty much all of the metabolism that we go through, acetyl-CoA is the keystone species. So whether you're doing glycolysis or if you're doing beta oxidation, whatever you're really doing to get your own energy, we turn that substrate into acetyl-CoA first, then pump it in the citric acid cycle, and everything kind of, becomes equal once it's acetyl-CoA and enters the citric acid cycle. But, all the way that we make energy, this is kind of like our juncture point. It's a big confluence of energy systems, so to speak. Acetyl-CoA is very important. You definitely want to be comfortable with it. All right. So now that we talked about it, let's actually go through the full process of the citric acid cycle and see how this stuff all connects. So I also just want to warn y'all, like some of these steps, we're going to go through fairly quickly. As we said in the very beginning, we are focusing primarily on big picture ideas like counting your carbons, word root, enzyme categories, rate limiting steps. That's what's highest yield for the MCAT. But there is some good stuff to memorize as well. And so we might as well give you all a little mnemonic, and the other teachers can put this in the chat as it comes up. But this is something that we do have from our review books. And so a lot of you might have seen this before if you've gotten the Kaplan books or if you've taken a class. But a little way to remember the order of the substrates of the citric acid cycle is people often say please, but remember, technically speaking, this includes pyruvate decarboxylation. So I'm going to start with just the C, but it's can I keep selling seashells for money, officer? It's like a little kid on the beach collecting seashells and trying to sell them to make some money. And then the cops come up and try to shut the poor kid down. And little kid's like, please, can I keep selling seashells for money, officer? Little way to remember the mnemonic if you want to envision a poor little child being yelled at by the police. But the thing is, cheesy mnemonics are usually the best. In fact, if you look at a lot of the mnemonics from medical schools, usually it's the dirty mnemonics that stick the best. And so if you are into such dirty mnemonics, feel free. They have been shown to be effective. We try to avoid controversial things when possible, though, when teaching. But it is up to you how you want to remember the things that you need for test day. All right, so what we're going to do here is not focus on just memorize these structures. As I said, what we want to do is focus on the big picture stuff, like what you can infer, like how they can connect connected questions via critical thinking, organic chemistry, rate limiting steps, and once again, enzyme categories will be a very big part of it. So we do get to introduce a new class of enzymes here with our first step. And this is when you bring together oxaloacetate and acetyl-CoA. And so this is an example of a condensation reaction, you can say, because you're bringing multiple things together to form new bonds. So condensation. And so now this is a specific enzyme that, notice, is going to synthesize a new molecule, and it does not require energy. Notice this is a spontaneous process. You don't have to put ATP in to make it work. It runs itself, which is why it's citrate synthase. This is going to be in contrast to synthetases. We'll see one later on. Those usually either require energy or produce energy themselves. Whereas if it's a synthase, it means no ATP involved. So basically, you're going to make this stuff for free. Doesn't take any energy, or at least doesn't take any ATP specifically. Cool. All right, so that's our new class of enzymes, right? Synthase. All right, we've got 2 carbon acetyl-CoA. We have 4 carbon oxaloacetate, and that's going to combine together to form citrate. So now you might realize, once you actually see the actual organic structure of citrate, why we call this cycle the TCA cycle or the citric acid cycle. Notice when you look at citrate, you've got, count them, 1, 2, 3 carboxy groups, hence the nickname, the tricarboxylic acid cycle, because really that's just another way you can call citrate. There's not many molecules that have 3 carboxylic acids on it. So if you say tricarboxylic acid, you're pretty much talking about citrate. Hence having the 2 names effectively means the same thing. So that's the source of the so many names. And of course, Krebs cycle comes from the scientists. So there's lots of names for this thing. It's kind of annoying, but you got to know them all because you don't know which way you're going to see it described in testing. All right, so there's our different structures there. And so all that red box is showing you there is it's showing you where the acetyl-CoA went. So yeah, your acetyl-CoA is being re-represented here via that structure. All right, that's the first step, citrate synthase. So once again, this occurs in the mitochondrial matrix, as does the entirety of the TCA cycle. It's a condensation reaction, and it is irreversible. This is actually pretty important that it's irreversible. It's gonna, well, not for this class, but for if you were to look up fatty acid synthesis, it's important because the way that we synthesize fatty acids is basically building them from acetyl-CoA. And so ideally, you would take your citrate, you'd reverse that reaction, but we can't do that easily. That requires energy. And that's basically it's going to come into play in the context of fatty acid synthesis, and that you actually need ATP to basically reverse this. Right? That's because it's irreversible as is. But of course, put enough energy in, you can make anything go backwards. All right, so, well, not anything, but you know what I mean. Well, theoretically, I guess they say that people actually undenatured eggs recently. Like, I don't know how that's even possible, but apparently someone figured out how to undenature an egg. So if that's true, who knows what's actually reversible that we didn't think was. Who knows? All right, so that's beside the point. So notice once again, we have the same idea. We've got a bunch of reactants. We'll speed it up. NAD+ and ADP would speed it up. Whereas ATP and NADH and other products like succinyl-CoA that you'll see later on will effectively slow it down. So you're going to see so many of these little green pluses and red minus signs. All of it's showing the exact same thing, though. It's saying that reactants increase the speed of reactions or will push them forward. Products slow down reactions, drive them backwards. That's just Le Chatelier's principle, classic Gen Chem stuff. That's just showing it in the context of biochemistry. All right, so that's our first step. Second step. So this is just an isomerization, as you can see, turning citrate into isocitrate. But there's a really cool thing you can do to think about this. And I want to show you the actual organic structures here to see why this actually matters. So this is a picture of isocitrate. Notice you have a tertiary alcohol, sorry, citrate. And this is isocitrate, which is a secondary alcohol. So what's the benefit? Why would you want to turn a tertiary alcohol into a secondary alcohol? And one thing I challenge you to think about here is, remember, every bit of this carbon, every single one of these carbon atoms is going to eventually become carbon dioxide. That is the net product of our metabolism, is basically breathing out CO2 and water. So we make water and CO2. That's our products, as well as, of course, energy for ourselves. So the closer you can make these carbons to CO2, the further down the line you are. And a tertiary alcohol cannot be oxidized. A secondary alcohol can be oxidized. And how much can you oxidize a secondary alcohol? So remember, primary alcohols can be oxidized all the way to carboxylic acid. Secondary alcohols can only be oxidized to ketones. And so this is going to get oxidized to a ketone. Now remember, to go even further, that if this is a carboxylic acid, it means the next door carbon could be called an alpha carbon. The next one would be called a beta carbon. And so the reason that I bring that up, though, is that if you are doing this so you can oxidize the alpha carbon, it would make sense for the next structure to be called alpha keto something, and that's exactly what it is. So via making the secondary alcohol, we can then go through the next step and actually oxidize that secondary alcohol into a ketone, hence alpha keto glutarate. So it's another oxidation reaction. So that's kind of the cool stuff. And that's what matters most, I'd say, for the MCAT. Not memorizing the structure of isocitrate, but if you were to be given a picture of it to infer, like, oh, cool, this can be further oxidized, whereas citrate, where it is, you can't oxidize it as it is. And so that's the kind of stuff that they love you to think about. All right, so that's the idea. We're going to form alpha ketoglutarate and some carbon dioxide. Also, just as a little bit of a shortcut, generally speaking, if you see a reaction that produces carbon dioxide, it is going to be an irreversible reaction. So most of the, especially in Krebs cycle, that's always true. But for many reactions, they do tend to be. It's not everything, like not things like, well, I won't get into it, but not everything, but tends to be true. And so you're going to make carbon dioxide. That carbon dioxide is going to diffuse away. That's the same CO2 that we're breathing out all the time. It's a product of our sugar consumption. All right, and so yeah, that's kind of the main idea there. We have isocitrate. It's notice a dehydrogenase because we're going to be making NADH. Anytime you have an enzyme that makes NADH, it will always be a dehydrogenase. And then, of course, we're also going to decarboxylate it, making your alpha ketoglutarate. All right, so once again, this occurs in the mitochondrial matrix, just like everything else in the TCA. It is the first step that generates NADH. It's the first step that generates CO2. And there's one other very important thing to know about isocitrate dehydrogenase. And the fact that I said it was a very important other thing to know about isocitrate dehydrogenase is a hint that this is actually the rate limiting step. But RLS standing for the rate limiting step of the TCA, otherwise known as the CAC, otherwise known as the citric acid cycle, otherwise known as the Krebs cycle. Once again, it has lots of names. So that's the rate limiting step. Definitely worth knowing. All right. And as you said, it is irreversible, right? Often and always in the TCA, if you form carbon dioxide, it's going to be an irreversible step. The next step will be the same. And other than that, same stuff as before. A bunch of reactants, speed it up. A bunch of products, slow it down. That's nothing new. You've seen this a dozen times. It's the same thing. Doesn't, it's just all products going to slow things down, reactants speed things up. All right. So notice next step. And so just like a guess. So what do you think the name of this enzyme would be if it's something that is going to, notice, produce NADH? This is the stuff that matters most here. It's just inferring the names of these things. And if we know that we're going to be having creating a new reducing agent, right? High energy electron carrier, it must be another example of a dehydrogenase. So you got it. This would be, once again, alpha ketoglutarate dehydrogenase, because we're going to produce, of course, NADH. And once again, it's a complex because we have a lot of stuff going on. You're producing CO2, lots happening here. But once again, mostly for us, it's a dehydrogenase. It's the second step, which means that we are now down to 4 carbons, right? So remember, alpha ketoglutarate was 5 carbons. This means succinyl-CoA is 4 carbons. And we basically made CO2 and NADH. Biggest takeaway for you all, though, remember, it's a dehydrogenase, which means you're making your high energy electron carrier, not from scratch. It's just that you're reducing NAD plus into, of course, the reduced form NADH, right? So NAD plus is the oxidized version, NADH is the reduced version. All right, as we said, we made CO2 and NADH, and we're down to 4 carbons. All right. Cool. And once again, same thing as before, bunch of reactants, speed it up, a bunch of products, slow it down. So nothing new there. That's all the same. All right. And we showed this before, the connection, how this also slowed down citrate synthase. But once again, products, right? This is a full product of the first step. So it's going to slow down upstream reactions. So nothing new there. All right. Now, the next one, we got 2 more that are interesting to talk about. And so the next one is one of the interesting ones because you make GTP. So now the difference here is that we're not directly transferring the phosphate group in the same way. So it's not a kinase, but it is an enzyme that, remember, we said synthases don't require or produce energy. Synthetases do. And so this is the example of the synthetase. So it says succinyl-CoA synthetase. And so that's the difference here. Notice we're actually grabbing, the reason this one's different is we have our inorganic phosphate. We're not transferring it from an ATP directly. That's what we did with all of our kinases. In this case, it's inorganic. So it's a little different, not a kinase, same idea, but in this case, it's a synthetase. It's slightly different. And once you make this GTP too, that GTP gets converted right to ATP afterwards. And so that's, don't worry about that. But you can think of it as basically making ATP, even though technically it's GTP. That GTP gets converted into ATP so we can use it. All right. So once again, everything in the TCA happens in the mitochondrial matrix. This is another example of substrate level phosphorylation, just like we saw in glycolysis. And you can also, once again, we showed the ATP already. And that's going to form your succinyl-CoA. Exactly. So you had the energy there you got from the thioester bond in your succinyl-CoA. So notice we have lost our CoA group, right? So CoA group is now gone. We're down to now just succinate. All right. So one more thing. And as we said before, that GTP is generated directly. It's the only step that you make GTP directly, but of course you do turn it into ATP. So now succinate is a really important reagent because it is actually another part of the, well, we'll get to this in a second here. But succinate's pretty interesting because this enzyme here, succinate getting turned into fumarate, this enzyme occurs not only in the Krebs cycle that we see here, it also happens as part of the electron transport chain. So this is actually really cool. We don't cover it in today's class, but we do it later on in other channel classes. But this is actually, do you guys know what complex it is? It's known as succinate dehydrogenase. You guys know what complex it is? So remember how we said NADH goes directly to complex 1? This succinate, this is the step that you make FADH2. That's what actually goes to complex 2. So succinate dehydrogenase, this one is the only one that is not actually floating around in the matrix. It's bound to the membrane because if it's part of the electron transport chain, the ETC is on the membrane, which means that it's basically got to be in that location. So, right, of course, it's, stuff in the matrix reacts with the membrane, it's touching it, but it's not free floating like the rest of it. So this one's really unique. And so basically it is the second substrate, right? It's the complex 2 of the electron transport chain, otherwise known as succinate dehydrogenase. We cover that in future classes, not this one. And that is going to basically make your FAD. And yeah, so basically the FAD is covalently bound. It makes FADH2. FADH2 then gets oxidized and keeps pushing electrons down the chain, which is what happens in the electron transport chain. So yeah, this is a really unique one. It's the only one that makes FADH2. It's your only source of it. It's the only one in the citric acid cycle that's also part of the electron transport chain as well. Cool. All right. Cool beans. All right, let's do the next step. So next step, fumarate into malate. As you can see, there's nothing super exciting happening here. This is kind of cheesy, but a little way that I have to remember this. Like, you don't have to memorize this step. It's one of the least important steps. But if you are getting down the system, if you already did the mnemonic, you got it down, notice all you do here is you add water across a double bond, because if something is fuming, right, fumarate, like it's on fire, you got to throw water on it. So that's what we do to get rid of the fumarate, put out the fire, throw some water on, and that turns into malate. But in reality, the orgo that happened here is fumarate has a double bond. You add water across it, turning into an alcohol. So fumarate, of course, has an alkene, whereas malate, of course, has an alcohol. But that's kind of beyond the scope of what we care about here. Most importantly, it's just another step of the Krebs cycle. You're, basically just going further down the line. Be aware of where it is in the context of the Krebs cycle. That's mostly it. But if you want to know, adding water across the double bond. All right, and that brings us to our last step of the Krebs cycle, otherwise known as the citric acid cycle, and that is turning malate into oxaloacetate. Any guesses what enzyme this would be called since you're making NADH? Yeah, so it's another dehydrogenase. So pretty much anytime that you're going to form NADH as one of your reagents, it's going to be a dehydrogenase. This is no exception. So basically malate getting turned to oxaloacetate. Remember, and I know I mentioned this before, but the big picture here is oxidation reduction. Since you are going to reduce NAD plus into NADH, that means we must be oxidizing malate into oxaloacetate. So that's like a real kind of common theme that you'll see in a lot of this stuff. It's, in terms of trends, so like if one thing gets oxidized, the other things got to get reduced. All right. So that's malate dehydrogenase, as you'd expect. Once again, mitochondrial matrix, just like everything else in the Krebs cycle. Of course, remember that your succinate dehydrogenase is unique. It's still in the matrix, but it's bound to the membrane in the matrix. It's a little different. This is classic standard stuff. Makes an NADH, as we said before, as well as it's the last step. So yeah, last step of the TCA. And so remember, this is the Krebs cycle, or the citric acid cycle, which means that your final product then is also the first reactant. And so it's going to then, of course, combine with acetyl-CoA, go into citrate, and then the loop continues on indefinitely. All right. So that's it. So what we'll do is we'll do a couple summaries here. We'll do a couple questions. Then we'll have Petros jump back on. He'll be the one that gets more into the test, the strategies. We were talking about scheduling. I guess not scheduling, but like big picture stuff, like action items, what you need to do from now to test day. But we got at least another 10 minutes or so before we get there. All right, so let's do some summary stuff. So as we said before, in all of these steps, if you are ever going to talk about control points, the products, ATP and NADH, that's a sign that you have enough energy. And if you have enough energy, you are going to slow everything down, right? So it's going to slow down your, pyruvate dehydrogenase complex, right? That's the only complex in that step. It's going to slow down, of course, your citrate synthase. It's definitely going to slow down your rate-limiting step of isocitrate dehydrogenase, as well as everything else as well. So that's the idea. Whereas if you don't have enough energy, that means you've got a lot of ADP or AMP and NAD+, then you're deficient, which means speed up every step that can use it. And so you're going to speed up pyruvate dehydrogenase complex. Remember, that's its own enzyme in its own step. So like that is its own rate limiting step because there's only one step in pyruvate decarboxylation. It's also going to speed up the first step here, citrate synthase, as well as, of course, your rate limiting step and your other irreversible step there too. So that's the main idea. Notice that your irreversible steps here, these are the ones that are going to be producing CO2. And of course, definitely know this is your rate limiting step. Definitely know that is your rate limiting step. As I said before, super high yield stuff there. All right. So let's talk totals. So we talked about control. Let's talk about what we've actually made here. So if we count up everything that we did, oh, sorry, here's a couple more feedback, right? We mentioned this before, but remember, negative feedback is very common. Don't stress about the specific relationships there. Do know the feedback mechanisms. And remember also, just big picture, if you have calcium, calcium's a sign that you're in need of energy, right? So calcium shows speed things up. So that's why we're focusing, of course, on your rate limiting steps. That when we go through glycolysis, you get a total of 2 pyruvates. We have the 2 there, pyruvate decarboxylation, as well as 6 NADHs in your TCA, as well as 2 FADH2s, and 2 GTPs. All right, so that's kind of the summary here. So the net stuff that we used up 2 pyruvates, we made 2 NADHs. Oh, sorry, this is the stuff that we used. Well, we actually, oh, sorry. This is actually, I take that, I apologize. So the top box here, this is all showing your pyruvate decarboxylation. So remember, there's 2 different steps that happen here. So pyruvate decarboxylation is the top, as well as the bottom here is all of your Krebs cycle. So we used up 2 acetyl-CoAs, we made 6 NADHs, 2 FADH2s, as well as 2 GTPs. Cool. All right. So that's all the summaries. We like to end classes with questions. In fact, one thing I want to say too, just about big picture, MCAT prep stuff. Whenever, like, my number 1 most important piece of advice for everybody is do not just study for the MCAT. Like, I know you're going to have to study. We often don't know a bunch of the content yet when we're getting ready for this exam because it tests effectively everything you learned in your science classes in college. But what's more important than studying is practicing. So the biggest change I would say that every single person here can easily incorporate is anytime you ever study for the MCAT, like anytime you have a session, like maybe you're going to study for 2 hours. At the end of that session, end with questions. Never just read a book and say, cool, I learned stuff. End with application, end with applying it, end with questions. Ideally, if you have the time, try to end with passages, but definitely end with at least a couple questions, which is what we're going to do right here. All right, so let's give it a shot. So same thing as before, Chris, if you don't mind grabbing that poll, we're going to have you guys answer this question. Once we got some answers, we will discuss. So there's one, what's the enzyme that they're focusing on here? So aconitase is the big issue here, right? So if we lack aconitase, notice that the answers are all different enzymes of, well, it's all different substrates specifically of the Krebs cycle. So these are all different substrates of the Krebs cycle. So we want to figure out aconitase. So if you lack aconitase, remember you have some kind of reactant, that's going to give you some kind of a product. And so what we want to do, well, I guess let's go big picture here first. So what will you have too much of, reactants or products, if you lack this enzyme? So if you do not have an enzyme that catalyzes this product, what that basically means then is that, let me change my pen color here. If you cross this thing out, you're going to have a low amount of products and a high amount of reactants, which means we got to figure out what is the reactant of the aconitase enzyme. And as we're kind of split in the polls here between 2 of them, but we're basically split evenly between succinate and isocitrate, it looks like. And so as we said here, we are not going to have any isocitrate. So remember the reactant here to actually give the full structure here. This was citrate itself. This is what aconitase does. And the product, of course, is aconitase. All right, so that's the idea is basically if we lack aconitase, oops, we're not going to have isocitrate. Sorry, I wrote aconitase twice, but that's isocitrate, which is what the correct answer is there. Succinate's a little further down the line. Yeah, so succinate, malate, and fumarate, those are all later parts of the Krebs cycle. Isocitrate, that's right in the beginning. That's right after aconitase. So what you could do too for this one, if you know aconitase is in the earlier parts of the cycle, just choose the substrate that is also earlier in the cycle. It'd be a shortcut way to answer that question. Cool. So let's do another one. I think we got 2 questions here. All right, same thing as before. We'll bring up a poll for you guys, throw them in, let us know what you think, and we will discuss. Okay. Cool. So we're mostly going for, I like it. We're mostly going for A and D. So nice work. So now the reason that these are nice answer choices here is because nice moving more towards D. So yeah, these are main answer. I'm looking good. So the idea is that this is the Krebs cycle may have evolved from this pathway. So based on the structure of the modern Krebs cycle, what's probably true about this pathway? And so the idea here is turning succinate into fumarate. That was, of course, our succinate dehydrogenase that made FADH2. We talked about that before. Fumarate to malate, that didn't take any energy. Malate to oxalic acetate, that made NADH. And so there's a little split here. So I guess if we look at this backwards, there's a couple ways we can actually think about this question. Oops, sorry. But the idea is if you break down each step. Now, I agree that what you guys are doing here, it's totally true that if it does not consume ATP, it produces NADH. That is the TCA. That's what we're doing. If you go through these actual steps themselves, though, like you guys are thinking the right thing saying A and D. As I was saying off the bat, that makes sense. That's the Krebs cycle. That's TCA big picture. But if you go through the individual steps here, you have oxaloacetate making that into succinate. In that pathway, notice that we need NAD+. So in order for that reaction to occur, you need NAD+. Let's get a connection to this. So one of the things that this relates to, and you guys were definitely right going for C and D too, because remember, we didn't consume any ATP in the TCA. So another way you can go through this is right off the bat. In the Krebs cycle, there's no consumption of ATP. Get rid of A and B right off the bat, but nowhere in the cycle. But as you go through the entire Krebs cycle, many of these reactions require NAD+ to function. So if we go back to our example in glycolysis, remember that, well, actually, we didn't mention this, but you turned NAD+ into NADH in the middle of glycolysis. And so if you are doing anaerobic respiration, that's why fermentation happens. The reason that we ferment if we don't have oxygen is to actually turn that NADH back into NAD+, because without NAD+, you can't do glycolysis. This is a super hard question because what it's doing is effectively the same thing. In order for this reaction to occur, you need NAD+. So this would, of course, then in its step, basically produce NAD+, so you can then further the other reactions. Super tough question. Honestly, I would not worry about really complicated questions. What I would work on is eliminating wrong answer choices. And so the fact that you can get rid of A and B right off the bat, because you know nowhere in TCA do you ever use ATP, get rid of those two. So then you want to think, all right, NAD+, NADH, it's going to be one of the two. And when you break down the steps to get there, you are going to, of course, need NAD+. That's a really hard way to end class, actually. I apologize for that one. But we're not done yet. We actually got a lot more to talk about. Petrus is going to be up with you in a second here. and get more back into the idea of what do you need to do? What are the steps? How do you prepare for the MCAT, not just to get ready for the test, but efficiently and effectively? And that's what we're here for. So let me know, Petrus, whenever you're ready. Once this camera has jumped back on, I will hop off mine and we will do the smooth transition that we did before.
Thank you, John. And I'm going to share my screen. So, what do you guys think? I mean, it was pretty awesome, right? A lot of good stuff as far as metabolism is concerned. And really, if anything, you've now seen all the things that you need to start thinking about in terms of just one topic, right? One area. There's a lot on this exam. And as you saw, there's a lot of stuff that's kind of interconnected, right? In terms of the materials, in terms of the different science that's going on, even in a very, very specific system and systems like we looked at here. But at the same time, it's all interconnected, right? Because the rules for oxidation and reduction that we looked at here today with metabolism apply in other places, right? And there's, they're going to be kind of cross-functional in many, many ways. So as we said, this was a snapshot into the Kaplan classroom. This is a lesson that actually both John and I and Ari and Chris and Kevin, who are all here today, that we teach on a regular basis. I think John, you taught it last week, right? So it's part of class. But really, what it comes down to is it's a long road in terms of your preparation and in terms of what it is that you need to be doing to get yourself ready for this exam. So a couple of kind of broader, bigger scope kind of ideas, things to look at is, number one, make sure you know where you're starting at, and not just necessarily in score, just really kind of as far as your skill level, as far as content areas, really important to make sure that you very early on identify where your areas of opportunity are as far as raising your score is concerned. I mean, you don't raise your score by not learning new information, by not taking on a challenge. I mean, that's really what it comes down to is, where are your areas of opportunity? Where is it that you're struggling? Where is it that you need to be putting focus on? And then making sure that you're putting the appropriate amount of focus on those areas. And I'll give you an example of that, of what I mean by this. Something like metabolism that we saw here today, pretty high yield, right? It's going to be a topic that is going to be tested consistently on this exam. It's going to be tested a lot. There are going to be a lot of questions that will kind of revolve around the information that you saw here today. But something like alpha and beta decay, a little bit lower yield. So you don't want to be spending an equal amount of time on all subjects, and you don't want to be spending an equal amount of time on all topics. One, because that's the nature of the exam, but two, also your individual strengths and weaknesses, right? Things that you're already really good at. You want to be focusing on areas where you can actually see your score improvements. So kind of knowing your starting point, really, really important, something that we emphasize a lot with students. And I say this to all my students, like really know kind of where you're at right now, but then also have a goal, right? Have a target score, a goal of where it is that you want to get to. But what's important to realize is that studying for the MCAT is very, very different than the studying that we've been doing. It's not about reviewing your old textbooks. I'll tell you right now, there are many, many more topics that are in a college textbook than will be tested on the MCAT. The MCAT has a very, very finite kind of set of topics and content areas that you're responsible for. So one of the first things that you need to make sure that you're doing is actually building out a study plan and following a study plan that follows the blueprint of the exam. And that's actually how our course is built. We build our course based off of what you're going to actually be tested on. So you're not wasting time, because we all know that time is pretty valuable. There are all sorts of things that we'd rather be doing than studying for the MCAT. So making sure that you're actually putting emphasis in those right areas. At the same time, as you saw today, it's not just about knowing content, but it's really about being able to apply the content and apply your critical thinking skills into these questions to be able to reason out an answer. In fact, that is the core skill that is being tested on the MCAT, is your ability to actually apply this content and reason out an answer choice using the information that you have in front of you. And so a big part of your preparation is going to be learning strategies and, more importantly, doing drills and practice that you are going to kind of strengthen those skills. The questions that you saw here today, the questions that you'll see as you're practicing for the MCAT, those specific questions will never show up on your actual day of the exam. But the skills necessary to answer those questions, the strategies necessary, the content necessary to answer those questions will show up over and over again. So the earlier you're able to kind of incorporate kind of that overall approach is going to be much better for you in the long run than if you just try to review content and only have content be part of your preparation as far as this test goes. Another kind of top tip for success here, getting access to at least 6 practice exams, super important. The reason for that is that, as I said at the beginning, it's about building endurance, right? It's about being able to execute and to endure the length of the exam. And of course, along the way, tapping sources of expertise so that you make sure that you are doing the things that you need to be doing in order to be, in order to be successful as far as this test is concerned. So really important to kind of be thinking about all these things as you're working through this material. Now, we of course have full courses, right? So we have full courses that are, that include the live instruction like what you saw here today. But sometimes you just want to maybe do some questions, maybe you want to just watch some videos, want to get some test-like questions, and then maybe there's, of course, later down the line, there's the need for actually putting together your application. So we have a couple of newer educational resources available for students, which I wanted to tell you about because I think they're actually kind of cool. If you're looking for just lots of kind of drills as far as practice questions are concerned, we have our adaptive Q bank. What the adaptive Q bank is, plain and simple, is about 2,000 or so MCAT questions that we've curated, but then it has this really, really cool technology that actually adapts to what your individual needs are. So it's not just individual needs as far as the content that you need to work on, but also the level of difficulty of the questions. Let me kind of put this to you this way. We'll use the gym analogy. If you've never worked out and I say, okay, here's a, 250-pound weight and I want you to lift it up, you're not going to be able to lift it up, right? You're not going to have you're not going to build the muscle in order to be able to lift up that weight. Unless you have a really long lever, of course, but that's a physics problem. But you want to kind of start at a level that you are comfortable at. And then as you get better and better, you want to give yourself more and more of a challenge. The MCAT's the exact same way. You don't want to be tackling super hard questions early on because you're only going to get discouraged. So what the adaptive Q bank does is it's always kind of giving you questions that are in this perfect zone, kind of that sweet spot where you're not only getting challenged, but you're also able to learn from the experience and then improve your performance to get harder and harder questions. So the adaptive Q bank is something that you can get on its own. A lot of students just want questions, and so that's one way to get questions. It's included as far as our full course is concerned, but something that you can also get on its own if you'd like. We have practice packs, and what these practice packs are, there are different packs. You can find them on our website. If you just want full-length practice tests, or you just want science review videos, so not live instruction, but just recorded instructions. So get videos, that just break down individual topics, or you want passage-based question sets, so that you can actually start to get some practice in terms of the way that the test is going to be testing you on. We have practice packs available, which is something, that some students, that's just what you're going to need and something to look at. And then, later down the line, and I'm talking like June, because you don't want to put the cart before the horse. We do have plenty of admissions consulting available. So you have something called Med School Admissions Help, which is help in either the personal statement or mock interviews. There's a self-paced admissions help. And then you can also get one-on-one consultation with one of our medical school admissions consultants. So that's something maybe to think about a little bit later down the line. Let's take care of the MCAT first, but something that is there for you as you look at it and as you're kind of thinking about what your future months are going to look like in terms of getting ready for this test and for the application process. The complete self-study bundle, if you are looking just to self-study, contains all the things pretty much that are on the screen here, except for the med school admissions consulting. So it's all the self-study practice, as it were. But I know, and I know this from the chat, because I saw a lot of questions coming in as far as like, what actually happens in classes or what's the structure of the class. So what happens in classes is what you saw here today. But as far as the what it means to take a Kaplan class, plain and simple, you're going to learn content. You're going to get lots and lots of practice. You'll have all the books that you're going to need, and we guarantee that your score is going to improve. So bottom line, it's a fully kind of comprehensive program, a program that really you don't have to go and get anything else. I mean, we get this question a lot of, well, do I need to go buy the books? No, it comes with the course. Do I need to go get the materials that are available from the AMC, from the people that actually write the test? And the answer is no. We actually include those as part of the Kaplan program. So there's literally nothing that you will have to go out and get if you are enrolled in one of our classes. When you're enrolled in one of our classes, we basically take care of everything. And really, it all works as a very kind of connected ecosystem. And I know that's a very kind of maybe a high-level word as far as describing this, but it's all very interconnected in the sense that as you're working through the material that we have, we are constantly analyzing your performance and actually have another personalized recommendations engine, so another technology that we use that actually tells you, here's what you individually need to work on. So pretty much you can kind of get rid of the guesswork of having to figure out what it is that you're going to have to study and just, do the studying. And sometimes the hardest part about studying is actually figuring out where it is that you need to be putting your focus on. And we essentially take care of that for you. Our Plus program, for those of you that might want a little bit more personalized attention, the Plus program includes 3 hours of one-on-one coaching with one of our MCAT experts. So it's essentially like getting a personal trainer. So, in addition to the personalized study plan you're going to get from the recommendations engine, you work one-on-one with one of our MCAT coaches to actually, build out that study plan and making sure that you're staying on track with that study plan and making sure that you're doing the things that are ultimately right for you along the way. Now, what you saw here today was one session out of over 90 MCAT channel sessions. So the way the course is designed, you have your core sessions, which is about 36 hours of class, and then you have over 90 hours of class that you pick and choose the areas, the topics that you want to ultimately, the lessons you ultimately want to go to. So essentially what this does allows you to customize your preparation. And most students go to all the lessons, but bottom line, if hey, I'm a rock star at thermodynamics, don't come to the thermodynamics lessons. The teachers that teach on the MCAT channel, both on camera and off camera, are literally our highest rated teachers here at Kaplan. So it's a very, very select group of folks. Everybody who's here today does some work on the MCAT channel. So you already know us, right? You've already met us, or met some of us, I should say. But the way the MCAT channel works is that it's on 6 days a week. We take Fridays off, but it's on 6 days a week. There are live sessions. You essentially, the sessions rotate as far as the different topics are concerned. And then you can pick and choose which lessons you want to ultimately go to. And so this goes from foundational concepts all the way up to the more advanced and, of course, the high yield content areas that are on this exam. And then we have them all recorded, so you can always watch them. If you missed one or you want to watch one a second or third or fourth time, you can either come again live or you can watch it recorded. But the bottom line is it's a very, very kind of flexible opportunity for you to be able to personalize your preparation and, again, do the work when you need to be doing the work so that you're not sitting here going, should I be doing this or should I not be doing this along the way. I mentioned this earlier, but the importance of getting practice and really getting realistic practice is going to be absolutely fundamental to being successful on this test. So I can't emphasize that enough. It's it's one thing to just read material. It's another thing to really get your hands dirty and actually get practicing with this material. And so you're going to get over 10,000 questions with our program. It's going to be well more than enough than you're going to possibly need. I saw a question come in earlier, like, how many questions should I do? And the answer is not 10,000, but 10,000 are available to you. So there's a nice variety as far as topics are concerned. And you can see on our website kind of what is available to you as part of our courses. And that's really what I want to kind of emphasize here is we've got a lot of different options. And it does take time. And it does take some kind of thinking and coaching, maybe even to figure out what the right option is for you. So what I would highly encourage you to do is get in touch with us. Let us know how we can help you figure out what is right for you. So we actually have a team of MCAT instructors. They all work in Orlando, so they're and actually some of them are now more remote. We have a team of MCAT instructors who are our MCAT advisors for students like yourself who just need to figure out what you're going to need to do in order to get ready for this exam and what the right path is going to be. For some of you, it might even be you're going to need private tutoring. Some of you might be, okay, I just need some questions. So we'll help you figure that out. So you can either schedule a time for one of them to give you a call, or give them a call at 1-800-CAP-TEST. I know that sounds a little, or you can even chat us if you wanted to, but calling sometimes is better for situations like this, so you can actually have like a full conversation. I know that's a very foreign concept these days, right? We rarely call each other, we text each other, or we chat, right? But this is one of those kind of life-changing moments where it's probably good to have a full conversation with one of those experts. So I'd encourage you to reach out to us so we can help you figure out, what's going to be right for you along the way, because that's really what it comes down to. Is, do I want a live online class? Do I need an in-person class? Can I do it self-paced? Should I get private tutoring? What I will say though, and this is something that we're quite proud of, and you should rest assured, regardless of which way you decide to take one of our programs, the material and the instruction is all going to be the same. So don't feel like, oh, I need to take a class in person, and then maybe there isn't a class that's in person in your area, and you're like, okay, now I can't take a Kaplan class. No problem, take a live online class, right? The live online class, oddly enough, is actually, we found it to be more interactive, because you don't have to raise your hand and wait to be called on. You just ask your questions, and a teacher's going to answer your questions right away. And so, bottom line is, we'll help you figure out which of these is the right kind of type of instruction for you, or if maybe you just need some self-paced practice, right? It really just comes down to you figuring out what is going to be the right thing for you and for your path forward. And we're going to help you figure that out, is what it comes down to. So just something to kind of think about along the way. I mentioned the personalization that comes beyond the adaptive Q bank, that actually comes as part of our smart reporting system. What the smart reporting system does, it's very simple, very elegant, but really complex all at the same time. What it does is it actually analyzes your performance on every single question that you have taken, and it gets smarter along the way. It actually learns about you and is able to make better and better recommendations about where it is that you're going to need to place your focus throughout your preparation. You're going to be studying for the MCAT between 300 and 350 total hours. That's class time, practice tests, practice questions, watching videos. I mean, everything that you're going to do, reviewing your books, everything is going to be about 300 to 350 total hours. You want to make sure that you're maximizing that time, because that is not a small chunk of time. I mean, if you are going to study for the MCAT for 3 months and you want to get 300 hours in, that's like 20 to 25 hours per week. And that's a significant amount of time when you think about it. So, that being said, we want to optimize that time for you, because it's not just about the number of hours that you spend studying, it's really about the quality of the time that you're spending studying. So the smart reporting system is one way in which we make that happen for you along the way. This is a nice little chart that kind of shows the differences between the different programs that we have, whether it's self-paced or private tutoring or prep or prep plus. But all this information is available on our website. So, take a look, check it out, kind of see what's out there. As I said earlier, registration for the MCAT opens this coming Thursday. So now is really the time to be thinking, all right, what am I going to do about my MCAT preparation? What am I going to do about this, very, very important exam at this stage in my career? So something really to kind of be thinking about and to be kind of figuring out, sooner than later, because once those test registration dates open up, you want to start registering for the test, and then you have an actual day. You have a day that your test is going to be happening. For some of you, you might even be looking for something more than our traditional classes. And for you, we have our intensive program. So if you're looking for something extremely intense, we have a couple of different options that are available. We have our winter intensive program, which is going to be coming up towards the end of December. Essentially, with that, you get about 400 or 500 hours of instruction. There's private tutoring, there's coaching. It's full-length MCAT simulations. It's really a boot camp type experience. John, as I mentioned, is one of our directors for our summer intensive programs. Summer specifically is another time that we do this, but you would be looking probably more at the winter intensive program if you're thinking of prepping and taking the test in either January, March, April, or May. But if you're thinking of testing later, so you're thinking of testing over the summertime, then summer intensive might be a place to be looking. So again, lots of different options available to you. So I want you to really be kind of considering those. And as I said, reach out to us and we'll help you figure out what the right thing is for you along the way. All of our courses, bottom line, we guarantee that your score will improve or we'll give you your money back. So, it's an insurance that's there. Our goal is for you to be successful on this exam. So, if you are, if you're not ready to take the test, or if you don't, you take the test and you're like, okay, I want my score to go higher. You can use the higher score guarantee and repeat the course. And if for some reason your score doesn't improve, we, of course, we'll give you your money back. But, rest assured, you don't want to, we're looking for you, looking out for you, I should say, in this process. So, don't take a low score as a final answer in any way. So the higher score guarantee is there for you in that regards. But with that said, I'll leave you with these kind of words of advice. The MCAT, it's a hard exam, but it's an extremely preparable exam. And it's an exam that, there's a very, very strong correlation between the amount and quality of work and preparation that you do that translates very much into the score that you receive on the test. It's easy to get hung up on the number. The focus that I would say you want to place is how can I get more questions right? What do I need to be doing differently? What have I not been doing? What do I need to unlearn as far as the way I've been taking tests, the way that I've been attacking, studying, and relearn it in a more efficient and better manner, and really apply that along the way as far as your preparation is concerned, and as far as how it is that you are kind of reinventing yourself to prepare for this exam and get the best possible score that you can. So with that said, I wanted to give a big thank you to the entire teaching team that is here. A special thank you to John for providing what I think was a phenomenal lesson, and I'm sure you do as well. If you have any questions after today, feel free to reach out to us. You can reach out to us on chat. You can reach out to us over the phone. Lots of different ways. Visit our website. You can chat with us there even. So reach out, and we'll figure this out. So we'll figure this out with you as far as kind of what's going to be right for you along the way. So with that said, big thank you again, and I will take this opportunity to wish you all a very good night and the best of luck on the MCAT and the best of luck in your med school admissions process. And let us know what it is that we can help you with, and we will hopefully our paths will cross again, either in another Kaplan class or on the MCAT channel or wherever it may be along the way. Thanks everybody, and have a great night. Take care.